Hello, I'm Noboru Mizushima from the University of Tokyo, and I'm going to talk briefly about autophagy in mammals. Autophagy is a cellular process by which cytoplasmic contents are degraded by the lysosome. This cartoon illustrates the autophagic process in mammalian cells. A membrane sac elongates, bends, and encloses a portion of the cytoplasm. The resulting double membrane structure is termed the autophagosome. Next, the outer membrane of the autophagosome fuses with lysosomes, allowing lysosomal enzymes to enter and degrade materials inside autophagosomes. Finally, degradation products such as amino acids are delivered back to the cytoplasm and used for a range of purposes. In summary, autophagy is an intracellular recycling system. To visualize autophagosomes, we use the autophagosomal protein LC3 as a molecular marker. These are mouse fibroblasts expressing LC3 fused to green fluorescent protein GFP. Each small dot represents an autophagosome. Under nutrient-rich conditions, we can see only a small number of autophagosomes. However, during starvation, the number of autophagosomes dramatically increases. We can therefore see that autophagy is a highly dynamic cellular process. We have applied this method to study autophagy in whole animals by generating GFP LC3 transgenic mice in which all autophagosomes are labeled with green fluorescent protein. Using this mouse model, we can learn where and when autophagosomes are generated. For example, autophagy is induced in almost all tissues following 20-hour starvation. Autophagy is also induced in many tissues of neonates immediately after birth because nutrient supply from the placenta is abruptly terminated at birth. Autophagy induction can also be observed after fertilization. This is an oocyte several hours after fertilization. In addition, autophagy can be induced in some disease models such as acute pancreatitis. This figure summarizes the pleiotropic roles of autophagy in mice. Autophagy fulfills two roles within cells, recycling and quality control. The recycling function of autophagy produces amino acids and other nutrients. Recycling is important as an adaptation to starvation, which is the most fundamental and evolutionarily conserved function of autophagy, as well as the production of nutrients during very early embryogenesis before implantation. The second role of autophagy is intracellular clearance, which can be thought of as a form of quality control. This function is achieved by degradation of abnormal proteins, damaged organelles such as mitochondria, and even invading bacteria, and is important for the prevention of neurodegeneration and tumorigenesis. Although autophagy research has expanded exponentially over the last decade, there remain many unanswered questions at a fundamental level, and we also need to develop our ability to apply these findings to human health. There are examples of the future challenges and the directions in autophagy research. Firstly, measuring autophagic activity is critically important in both basic and clinical sciences. However, currently available methods are still complicated and difficult to use in whole organisms. Furthermore, we have no means to measure autophagy in humans. There is a huge demand to develop new methods that can be applied in humans. Secondly, we still need to understand the role of autophagy in humans, both under normal and disease conditions. The majority of our current understanding of autophagy has been obtained using data from model animals such as mice. However, mutations in autophagy genes have been recently identified in human diseases. It is expected that autophagy plays a critical role in humans to prevent disease. Finally, researchers are now determining whether autophagy could be a therapeutic target in human diseases. This possibility is being widely tested using model organisms. One approach is autophagy inhibition in cancer treatment. Indeed, clinical trials using autophagy inhibitors against malignancies have started. Conversely, researchers also expect that autophagy activation would ameliorate neurodegenerative diseases or even aging by clearing abnormal or toxic proteins accumulating in neurons. It is also important to identify diseases that can be effectively treated using autophagy modulating medicines.